leadership of Dr. Bastani, who I'm told is known to our students as Dr. B.
Hello everyone, today we're going to be sharing the results from our research over the past 2 semesters where we followed quantum dots and their applicability in cancer imaging. Medical physics is the application of physics processes and ideas in medicine. This can be seen in MRIs, X-rays, ultrasounds, and many other processes. Medical physics is generally split into 3 categories: diagnostic, therapeutic, and modeling and optimization.
Therapeutic medical physicists use radiation to treat diseases, and they also develop treatments for their patients. Diagnostic medical physics develops science-specific procedures to diagnose diseases, and in addition, they also review dosimetry information about their patients. Modeling and optimization uses diagnosis images to create a plan based on mathematical and computational science to enhance treatment for patients. Diagnostic imaging uses a variety of methods to assist in accurately detecting injuries or diseases. Within the branch of diagnosis, there are 2 main routes, radiation and optical imaging. Some diagnosis instruments include PET scans, CT scans, MRIs, and ultrasounds. Particles used within radiation and optical imaging include gold nanoparticles, fluorodeoxyglucose, and quantum dots. Optical diagnosis within the medical field has many uses, and multiple instruments within the field include optical coherence optical coherence tomography, fluorescence spectroscopy, and imaging. Quantum dots are used in optical imaging because of both their unique optical properties as well as their ease in being able to be tracked and seen by spectrometers and microscopes. They also have a higher advantage over other organic photosensitizers.
What are quantum dots? Quantum dots are synthesized semiconductor nanocrystals that can come in various shapes, sizes, and compositions. Because of their optical properties, they are very great for imaging. They can be used in fluorescent microscopy and spectrometry. Additionally, because they give off both light, some particles, and various rays, they are also good for radiation-based imaging. Depending on the diameter size of the quantum dot you're looking at, the emitted wavelength will vary. Smaller quantum dots, typically between 2 and 4 nanometers, have more applications in like biological and optical imaging, whereas larger dots between 6 and 8 nanometers are more used for electrical systems, such as LEDs or like solar systems. If you look at the image, you can see kind of what we're looking at when we're looking at pure quantum dots in different vials, and under there they'll give you the diameter size and the emitted wavelength. And one thing to note is that they are not always emitting like this. These are being excited, and that's why we're seeing the color.
So, similar to light microscopes, fluorescent microscopes work by detecting a sample from a stage. However, the fluorescent microscopes excite a fluorescent dye within the sample, allowing the sample to emit its own light. The light produced from the fluorophore dye is of a longer wavelength than the light emitted from the fluorescent microscope, and so the microscope needs to have a shorter wavelength to account for this. So for example, if a sample emits in the green light range, the microscope needs to excite the sample with a blue light.
With the use of the fluorescent microscope, we are able to look at A549 cancer cells injected with these quantum dots. You can see this in image 2 and 3. The top, we have cadmium selenoid quantum dots with a 20 microgram per milliliter concentration, and down here we have indium phosphate quantum dots with a higher concentration. What you're looking for here when looking for quantum dots is you want to look for the smaller, more intense dots on the screen, and it's a lot easier to see on image 4, whereas the cells are these larger, dimmer clusters. One thing to note is that when injected with quantum dots, not all of them are uptaken within the cells. And you'll see that like here versus like this guy, these 5 all have dots in them, and you can see that because they're a little bit brighter than these guys.
Unlike microscopes, spectroscopy follows the interaction between electromagnetic radiation and matter, typically measuring the intensity of light versus the wavelength. Spectrometers work by sending an energy source, usually a laser, to excite a sample. Our laser that we used was a 405 nanometer. A camera then at the end of the spectrometer quantitatively measures the intensity of the light for a given wavelength and graphs the data collected. The intensity and wavelength of the measured light can be adjusted with filters and slit widths on the spectrometer.
Like Emily said, with the use of the 405 nanometer laser, we are able to excite different samples of quantum dots. And this is what results from a spectrometer would look like. Mainly what you're looking for on this slide is just to notice that these 2 different peaks for these 2 different diameter size quantum dots are at different spots wavelength-wise, which is what we expect to see. In the first semester, we are primarily interested in maximizing the cellular uptake of the quantum dots within the A549 cancer cells. To do this, we analyze cell viability data from a number of different samples, all injected with different concentrations of quantum dots, and pretty much finding the concentration that is going to lead to a lot of cellular uptake without compromising the viability just for cells that we want to keep. This semester, we are more interested in analyzing the emissions from these quantum dots. We wanted to compare the quantum dots by themselves to the quantum dots that were injected into the cancer cells. And we are also interested in comparing the results from the fluorescent microscope to those from the spectrometer.
So with the fluorescent microscope, the optical properties of the quantum dots can be observed by the bright cells on the screen. The dim cells indicate that the quantum dots had not been absorbed or uptaken by the cells.
To compare the emission spectra of the quantum dots by themselves to those uptaken by cells, which was our first learning objective, we used a spectrometer to analyze a vial of those cadmium selenoid quantum dots at 20 micrograms per milliliter, and we did the same thing with the same quantum dots and the same concentration on a slide injected into those A549 cancer cells. When you overlay the data on top of one another, you can clearly see that the emission spectra from the vial of pure quantum dots is much more defined and intense than that of the smaller peak from the slide. We believe this could be due to the setup that's required to use the spectrometer on a slide. It involves focusing the laser onto a very small point on the surface of the slide, whereas with the vial, the laser shines through it and the light kind of disperses all throughout. Additionally, there is a decrease, or generally a decrease in quantum dot intensity once uptaken by the cells. So those are a couple possible reasons for this.
So in conclusion, it can be seen that there was a clear spike at the intended wavelength in both the quantum dots in the vial and the quantum dots on the cell slide. And while the characteristic spike indicates that there is a spike, there are unintended high readings at the front end and the back end of the spectrum that we can't explain at this time. There's a calculated 724.91% decrease in intensity from the quantum dots on the cell slide. We think this could be due to the unintentional readings or from the absorption or uptake of the cells, or maybe even the interference from the cell membrane of sorts.
In the future, we hope to have a better understanding of the results that we gathered from the slide. We'd like to apply what we've learned over the past 2 semesters to other nanoparticles or cells, and finally, we'd like to come up with some procedures to ensure that any research involving quantum dot spectroscopy or fluorescence microscopy provide accurate and reliable data. Thank you. Any questions?
purposes because they're absorbed better? Or is there a different?
Yeah, I think, so the smaller dots are more used in biological processes because they can like more easily be transported, and you can also put like a shell around them, so even if you do add stuff to it, it'll still be small enough to be transported around.
Thank you. Anybody else?
Does it matter what excitation wavelength you use for the dots? Like, you got different sizes and all that, or can you excite them with the same wavelength?
So you have to excite it with a wavelength smaller than the dot itself. However, you can use a little bit, you have a little bit of wiggle room with it, but it's usually like 50 nanometers shorter than what the dot itself.
But yes, the 405 nanometer laser was an intentional decision.
With your knowledge on quantum dots, and since we see a significant decrease in the emission of dots once they enter the cancer cells, do you guys have any idea of how to maybe like increase the intensity, or how that could be used in a medical setting or improved to detect cancer cells?
Great question.
Yeah.
So the dots are coarse, and I may not be understanding your question, so if at any point you want to ask it again.
Okay.
But, so the dots have a core shell structure. The shell is really what's important. It's what allows it to be aqueous, and that's what allows us to use it in biological applications. And with the shell, there are what's called ligands, which are kind of what you would put on the cells to go after certain cancers. So if you're interested in maximizing the uptake,
Or increasing the intensity once you excite the dots so that they shine brighter.
I don't think so,
Okay.
because, yeah.
I think we would just have to get a more sensitive instrument to read it, because with
Without altering the cells, I would assume
Yeah.
no, because the dots are going to emit how they're going to emit.
Can I answer?
Absolutely, yeah. Dr. B, go for it.
So if we are looking just to increase the uptake of the cells, there are multiple chemicals that we currently use, like VMSO, for example, kind of like opens up the membrane of the cells. But as for increasing the intensity, I mean, one way is like to have more quantum dots.
Right.
But like shifting the excitation source does not affect the intensity. However, like one of the issues that we had was like the setup of the spectrometer is like in a way that the slide is kind of like in the middle, and then the lasers kind of like hits just like a particular like area of the slide. So if we can kind of like broaden the excitation, so it kind of like covers more, that could work. However, we haven't done it, so we don't know. But
Right.
we can try.
For the future.
For the future. Yeah.
Yeah. All right, thank you, everyone.